Hello everyone! Today I'm very excited to make a presentation which is a commentary on David Sinclair and the Harvard team's accomplishments on age reversal. It's actually age reversal, going backwards to a younger time, using the OSK protocol. Um, so I don't know how I can emphasize this as much, but I really feel and think that this is perhaps the most important scientific discovery in the history of mankind. If this technology works as it looks very much promising that it will work, and fulfills its promise, this could be the veritable fountain of youth that man has been searching for since they realized that their lifespan was short. Trying to expand it, we want more time. Um, so let's look into this more carefully. All right, so this story really started with two Nobel Prize winners. One was John B. Gurdon. You see here written John B. Gurdon on the left, and it looks like he did a little bit of age reversal on his hair. I wish I could do that one of these days. Maybe. I don't really care. I wear this for the glare control. Um, and the other guy on the right hand side, Shinya Yamanaka, who they shared the Nobel Prize, and John B. Gurdon did the work 40 years before Shinya Yamanaka. And I forget the dates, but the Nobel Prize was something like 5 years ago, in that time frame. And they got the Nobel Prize for their discovery that cells in our body have the remarkable ability to reinvent themselves. They found that every cell in the human body, from our skin and bones to our heart and brain, can be coaxed into forming any other cell. In other words, you can take the cell and bring it right back to the embryonic stage. This process is called epigenetic reprogramming, and its potential for new drugs and therapies is vast. If neurons and heart cells are damaged by disease or aging, then cells from the skin or blood potentially could be induced to reprogram themselves and repair the damaged tissue. So the winners, John Gurdon of the Gurdon Institute in Cambridge, England, and Shinya Yamanaka of Kyoto University in Japan and the Gladstone Institute in San Francisco, made their discoveries more than 40 years apart, with the work of Shinya Yamanaka being the more recent research, as I said. So, and this protocol of bringing adult cells back to the stem cell at the pluripotent stage, where they haven't decided which type of tissue they're going to be yet, it is the foundational research upon which David Sinclair's team and the Harvard team built their actual usable epigenetic reprogramming to bring an animal back to a youthful stage, but not all the way back to the pluripotent stage, which is undifferentiated, would just be kind of a blob. And I'll have more to say about that. Now this paper by David Sinclair and his team is one of the finest papers I've seen. It's packed together so tightly that when you look at what you're going to take out to do a summary, and if you're going to do a good service to this paper, there's almost nothing you can take out. It's so lean and beautifully written. So what I did was I paraphrased everything, and it's good for one. You don't want to infringe on the copyright. The other thing is, when you paraphrase it and put it in your own words, and I like to tunnel down a little deeper, because I don't. It's not for peer-to-peer at the level of Dr. Sinclair and his team. I'm more interested in opening it up, and so that a more broader audience can understand what's going on. But why I'm telling you that it's so tightly organized and efficiently written by Dr. Sinclair is to explain that I have to go through everything, and it's like a computer code, the wording, and I have to get everything correct. So at times I'm going to have to read from it, which I ordinarily don't like to do. I like to extemporize. But this time that I have to be extra careful because it's so precisely written. So with that, let's look into it a bit more. I'm also going to do some what it could be said mnemonic neural coding at times where I introduce sort of silly images. Actually, the sillier the better. They have to be very, very visual and unusual and illogical to code it in your memory. So I'll do that because I'd like you to be able to just listen to this and go through the presentation with me. And at the end, if would be so much better if you walk away and you just magically remember all of these things, maybe for the rest of your life, or at least the key things. So that might be a lot of fun. So I hope you enjoy what's coming up here. So I got to move myself over a bit. Yes, sirree. There we go. Oh, I'm making myself small, which is good. The paper's the big thing. So let's look at this. Information from what may soon become the paramount discovery in the history of science, and I really mean that, as recorded in the paper see directly below. Okay. So reversal of aging and injury-induced vision loss by Tet-dependent epigenetic reprogramming. Now that's the title of Dr. Sinclair's paper. It even took me by surprise. Let me read that again. So this is the title of Dr. Sinclair and his team at Harvard's paper. Reversal of aging and injury-induced vision loss, so it's reversal also of the injury-induced vision loss, by Tet-dependent epigenetic reprogramming. So, and Tet stands for 10-11 transfer. So it's very abstract. I'm going to show you movies and everything in this presentation that we're doing here. I'm literally, well, not literally, but intellectually, I've done backflips and will continue to do every gymnastics to make these points stick in your mind and be easy and fun to watch. So let's just go through it here. So my purpose here is to present the paper more approachably for the novice and to expound on the implications of this extremely important work. The actual paper above is among the very best I have read, and I highly recommend that you also read it, the original, and then I have the link written there. So you can screen save and then write the link and go to that paper. You can probably look up the paper by the title and get there. So that's continuing. Dr. Yamanaka demonstrated that induction of pluripotent stem cells from mouse or embryonic or adult fibroblasts by introducing 4 factors. So there are 4 factors for Dr. Yamanaka's technique. And one is the OC3, OC4, and the others are called SOX2. The third is called c-Myc, M-I-C, and the fourth is called KLF4. So applying all 4 factors to living mammals has been shown to induce teratomas. No, it's not the tarantula in New Zealand. No, this is a teratoma. a rare type of tumor containing fully developed tissues and organs, or causes death due to abnormal tissue growth. So abnormal tissues, these things can have hair and teeth, kind of scary looking things. Anyway, we want to avoid that. So we cannot use Dr. Yamanaka's process right out of his recipe book. They had to make, the Harvard team discovered this. By the way, I'm making no technical contributions to this except to maybe definitely explain a few things at a lower, well, I don't want to say lower, but more detailed level so people can understand it, hopefully. So using the eye as a model tissue, Dr. Sinclair's team at Harvard demonstrated experimentally that expression of 3 out of the 4 Yamanaka genes, that's OCT4, the 3 are OCT4, SOX2, and KLF4 genes. So 3 genes out of 4. And if you look, I'm going to put this in your brain later. You don't have to worry about trying to remember it, because I think by the time we're through, believe me, you will remember this. Whether you like it or not, it'll probably be stuck in your mind, likely for the rest of your life. And if this is true, the rest of your life might be many multiples longer than what you think it would be now. So, continuing on here. So the 3 out of the 4, but not the c-myc, the c-myc factor. Now, I'm personally glad that one is not necessary, because I have a heck of a time, as you can tell, even saying it. So that one we don't need. So the 3, OCT4, SOX2, KLF4, resets youthful gene expression patterns and the DNA methylation age. So the methylation, we'll talk more about that later, but that becomes younger. Like when you measure it, it doesn't have the defects that accumulate with age. So it makes youthful gene expression patterns. So you get you can run faster, your skin is smoother, you look like a 20-year-old, that type of thing. But we're talking about mice at this stage. Resets the youthful gene expression patterns, and the DNA methylation age gets younger too, of retinal ganglion cells. So the ganglion cells are the little nerves at the back of the eye, the receptors, and then they go out and they form the axons together, form the optical nerve. And promotes axon regeneration after optic nerve crush injury. So they did that to the mice to produce an injury so they could see if they could fix it. And this is just a summary. Now, don't worry, we're never going to have to crush your optic nerve, because that's part of making the damage and then fixing it. But what for humans would only be fixing the damage, not making the damage. The epigenetic reprogramming, or what they call reviver, it's a very cute acronym, reviver, requires the DNA demethylases expressed from the genes Tet1 and Tet2. So DNA demethylizes. So in other words, it's going to requires the enzymes that demethylize the methyl groups, which you're going to understand this so clearly later in the DNA. So we need those expressed from the genes Tet1 and Tet2, indicating that DNA methylation patterns don't just indicate age, that they actually participate in aging. And we'll see that more as we go. Thus, old tissues still retain their structure, a faithful record of youthful epigenetic information that not only can be accessed, but was accessed for functional age reversal in the experiments performed by Dr. Sinclair and his team. So what they're saying here is a summary, and we'll say this a few more times. is that within your body's genome, there's a record that's preserved into advanced age. And this record has been, at least for mice, has been accessed and used to do like what I'll call a factory reset on the body of these mice, and promising to do it on humans too. We haven't got there yet. But to know that there is information that contains the correct methylation, such that you can undergo this 3-step protocol. It's 3 steps by 3 genes, but it's put into the same, you'll see this later, like in tandem, tandem truck, multiple and 3 in series. By using these 3 Yamanaka genes, OCT4, SOX2, and KLF4, they're able to access this information and do essentially what I call a factory reset to bring the body back to youth, but not back to the pluripotent step as in Yamanaka protocol. So I know this is a mouthful, but this is mind-boggling. I mean, to think that somewhere in the genome it stores a record of what everything should be, and you can access this and then recreate, like to take this, reverse and most of the damage in the cells so that they go back to a youthful form. That's just, it's astonishing in the largest sense of the word. I've never been so astonished by anything ever as this fact here. So in other words, when you actively undo methylation, such as by activating the demethylaser enzymes expressed from the genes Tet1 and Tet2, then more of the original youthful pattern of methylation is restored, similar to what we might liken to a factory reset of the methylation program. So let's go over some of the refresher information that will help make clearer some of the molecular dynamics. Now, molecular dynamics sounds very scary, but I'm going to keep it very light. And at this point, I'm going to go inside your mind, and I'm going to use your optical center of your brain and the linguistic center, and I'm just going to pull them together and form these ironclad associations that, like I said, you'll have trouble getting rid of them for the rest of your life. It's very simple. It requires, though, silly images. The more illogical, the better. So by the time, I hope, I'm not going to completely lose scientific credibility for the rest of time, because I'm going to talk really silly for the next couple of charts. At times, there's a background core of science that I'm going to be programming right into your mind. But I'm going to introduce the silliest things I could imagine in order to have your brain form those associations in a way that is very difficult for them, lose it. So I'm going to put this, program it right into your brain, so to speak, if you'll just let me do it. And you won't have to make any great effort or anything. I'm not asking for that. Just put up with me for the next few charts, and you'll have the fundamental background that'll allow you to see. I'm going to show a movie too, and will allow you to understand what you're seeing, and you have this magically have these terms in your mind and know what they mean. So let's progress with that. Here we go. So remember I said that he used 3 genes, the OCT4, the SOC2, and the KLF4. And I even showed it there in the previous slide here, SOX2. There's either 2 SOX2. And OSK here, I said, it reminds me of that book, I'm OSK, you're OSK. maybe by the time I'm through, you might think you're OSK or OK, but you might wonder about me. I got to be careful here. So anyway, so OCT4. Now, do you notice there's these giant? I want to tell you a little story. You were at the beach, and suddenly two of these giant octopuses came after you, and they had 4 tall legs. So what, I don't know why we're calling them octopuses, but because of the suckers and everything. But they're OCT4, not OCT8. Okay, so they came after you, on the beach, and that's how we begin this story. So that's at the beginning. Okay, there's OCT4, and you had to fight them off with your sword and chop all their 4 legs off. Yeah, then socks 2. Now notice these 2 socks. They on the stumps of these octopuses, you put 2 socks on them, the stumps. You're trying to save one of them, because what if you had second thoughts? What if they were species that they were the last 2 of their kind, Anyway, the blood was coming out, and its socks got all bloody and everything. And you're not sure if it worked or not, because they slithered their way into the oceans. Now, octopuses, it is a fact that they can regrow their limbs. Now, it really broke your heart, though, that you had to do that to save your life. And your heart, it like broke apart, or at least it felt like it. In your mind, it felt like a jagged like a cliff opening up. Cliff, like when you saw this word cliff with a big K, because see here, the cliff, it's like a zigzag, and your heart is like held together by like a band-aid. And see how the K's, they fit right in all the pointy parts and they got a big K on the top because it's a big point. Now you got a little K that just fits in there perfect on the bottom. So that's cliff, like KLF4, and the cliff is because of the 4 legs that you chopped off. So it's KLF4 also. So you have OCT4 and the 2 socks 2, and the KLF4, the cliff 4. And together they make the protocol OSK. And see, even on the K fits into that same cliff that opened up in your heart, held together by a band-aid. Okay? And this reminds me, this OSK reminds me of that book. Remember, I'm OSK, You're OSK by Thomas A. Harris there. This book reminds me of I'm OSK, You're OSK. And this thing on top should remind you also of OSK, the protocol. All right, so let's continue on. Hopefully that's starting to stick in your head. Now, a methyl group. What the heck is that? Well, it's actually, I drew this myself here. It's got 3 hydrogens attached to a carbon. I don't know if you remember that carbon has ability for 4 bonds. So it likes to have that. Get all its outer valence electrons fulfilled, makes carbon feel very good, fulfilling its outer orbitals. Anyway, the 3 hydrogens connect to 1 carbon, and then it connects to another molecule. And we'll show you just what other kinds of molecules it connects to. And this is a methyl group. And it's used in the genome to tell which genes to turn on and which genes to leave off. Now, in your body, your eye cell somehow, and this is another mystery, and we're getting closer and closer now to understanding it. Your eye cell has the same genetics as your toe. Or let's talk about your toenail, something quite rudimentary. Your eye, with all of its fanciness, has the same genetic information exactly as your toe, to begin with, your toenail. But the methyl groups tell your toe to read from the toe part of the book and become a toe, and the methyl groups tell your eye to read from the eye parts of the genetic book and become an I. And the methyl groups are key to that. Now, over time, the methyl groups get taken off by mistake, and certain things happen. And the eye starts to forget, what it is, and starts to become a little bit more like a toenail. And other parts of the body, you start getting hairs growing out of your ears, and other things happening as the body kind of gets mixed up. Now, the beauty of this, and what makes this paper so immensely important, is that there is a record inside the genome that also allows us to do what looks to be, and has for mice been, a fully survivable and health enhancing factory reset to put the methylation structures back to what they were at a youthful level. And that's, again, it's just so amazing. It's amazing. So I'm saying the TH in methyl stands for 3 hydrogens. And I would even recommend you spell it methyl. I'll tell you how to spell it for mnemonically. METHHHYL, okay? 3 hydrogens and a carbon. Okay, so and then there's another group. We're going to have more fun with this one because they gave it a hard name to find images for, but I get around that. The acetyl group. So it's basically a methyl group. You see on the right there. Here, I'll get my cursor on the right. Is a methyl group stuck to, and I'll show a picture, a carbonyl group, they call it. Maybe you want to make a phallic association or something with that oxygen there, and to allow it to stick in your mind. People, I don't want to go very far, but I'll do just about anything to make something stick in your mind. So you get the carbon and the oxygen sticking up, and the hydrogen hanging below. And they call it a carbonyl group. Well, you combine the methyl group with the 3 hydrogens to the carbonyl group, and you get an acetyl group. Now, why do they call it acetyl? Well, we'll go next here. Again, and remember, I'm programming it in your mind as we go. See that acetyl? Oh, I flipped it around. Notice? Let's go back. See, I had it that way. Now I'm going to rotate it with the oxygen and the carbon on the top, the carbonyl group. That's going to be the seat. And the 3 legs are going to be like a 3-legged office chair, right? So here we go. We flip it up like that. And oh my heavens, it's a seat. You could sit on that. So, and what do I want? A seat. Phonetically, it's acetyl. 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 Okay? So the acetyl group. Now, if you sit on it, you got to watch it because the oxygen is a bigger molecule than the hydrogens, which are small. So it's like a big round circle. Now you could sit on the oxygen, and you're kind of on a tilt because, or maybe I should do it that way to match what you do in the diagram there. So I don't know if you noticed, but it's reversed from when you present it, but details. So you could sit. You'd have to sit kind of like an uncomfortable position there, and you might, because the oxygen is a bigger molecule than the hydrogens, you might slide off of it and fall back. You might even hit your head. So acetyl, an acetyl group, acetyl, and it's made of the methyl group on the bottom there, and the carbonyl group on the top. And now we put them all together. There, where they look, you can see the methyl group, and you can see that it's where it is in the acetyl group. And then you got the carbonyl group. Now, carbonyl, I suggest you spell it with the O. Make the O gigantic. Carbonyl. So it does 2 things. The oxygen is bigger than the hydrogen, and it sticks out the O for oxygen. Carbonyl group has oxygen. The acetyl group is methyl group with a carbonyl group, and it's like a seat when you turn it over. But remember, you might slip and fall off the big oxygen. Oxygen, oxygen. Okay, so there we go, and let's forward here. So now we're getting close to some of the biological dynamics. And here's a DNA strand. It's nicely labeled here on the bottom. And you have an enzyme, I forget what it's called, but some like RNA protease or something. It's basically going to read the DNA and then split it apart, and on the top there, it's going to make the what do they call it? It's like the, hang on just a second, complementary, that's the word I was looking for. It makes the complementary strand with that they call riboxynucleic acid, single strand, okay? And from that, you can create a protein from that. And I should also say, because copyright laws, this image, wonderful image, is courtesy of the Scientific American magazine, a marvelous magazine that I'm not affiliated with. I just want to express gratitude for having this diagram, which I did not create. Now, I need to have a complete understanding for just about everything. Otherwise, my mind rejects it. It bounces off. So if somebody teaches me a subject and they just give me the surface, my brain is designed to be incapable of learning at that level. I have to understand totally, or I don't. So I start asking questions. This structure here, what makes it go? Now, it's true that at the body temperature, the water molecules bounce around among each other, and they go straight if they didn't hit something. They bounce around at an average speed of 1,460 miles per hour at human body temperature. Now this is almost 50% faster than the motion of the turning of the Earth. You know, it goes 24,000 miles in one day or something like that, which is you know, close to 1,000 miles per hour at the equator, the spinning around the Earth at the Earth's surface. These water molecules in your body are going almost 50% faster than that. So they're just humming, just bang, 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 like banging with such a vibrational frequency. It just goes crazy. So what's happening is this enzyme on those strands is being banged on all sides, but sometimes it goes forward, sometimes it goes backward. It would like to, but we'll talk about that. And from side to side, but it gets, you know, blocked. But what the net effect is, this enzyme will go whatever direction it can go. So what direction, why does it go whip? When you see the later movie, you see these things whipping along doing their work in one direction. What makes it go, and why does it only go in one direction? So I need an answer to that. So if we look at that molecule, you can see it had the enzyme. It has a hole at one end, small hole, and a larger hole on the other side. And there's 3 strands coming out of it. Coming out in this case is to the left. 3 strands coming out and 2 strands going in, where this DNA is going in. So that means that if you try to push this thing back, that small hole is going to have, going backwards, 3 loose strands trying to get through that hole. Can't do it. Going forward, it has 2 tight strands. 2 strands, that's less, tightly bound. It can go through. So in this fury of being bouncing with the water molecules and other molecules, a frenzy of impacts, it can only go forward. It can't go backwards. And so what we see is this thing whipping along. So almost all of the cellular dynamics is thermodynamic and electrical, you know, the electrical attractions. And things bounce around, and they have a particular outside envelope with positive attraction areas and negative attraction areas. and they try every combination just stochastically by all the bouncing around. When they find something that's complementary, you know, where the positive matches up with a negative charge on the other one and vice versa, and the surface contour of the charge envelope, they stick. So if anything, that's basically how this system works. And I'm going to show you a nice movie, an amazing movie, which I did not create, but has the credits in it. Be sure to take that into account. The credits. Here's a DNA strand, and you can see that there's these histone groups. Histone groups, and they got a histone tail. And the DNA strands wrap around these histone groups. They're like spools. I call them histone spools because that's what's happening. And see the methyl group, the top there, but see those little red balls? They represent methyl groups, and they look more like what I have shown above there than the methyl group that we derived. And the acetyl groups, remember the chair, acete? They're the green little spheres, and they have different functions. Notice when you put the acetyl group in there, it actually lowers the charge on the histone tails, and so it allows the spools to unravel. And this basically, the acetyl group, when they're put on the histone tails, they cause they diminish the charge with the attractive charges, and they allow and cause the spools to unravel. And so the DNA is exposing itself, and then it can be read by those RNA enzymes that produce RNA strands, complementary strands to one of the strands of the DNA. And these histones, where they're located, they're almost evenly distributed down the DNA, and they have their own little match where they bond to the DNA. So they have their own electromagnetic contour that matches with the DNA contour. And nature's done it so that they plan, so like the more or less even spacing in the DNA. So you get a histone group just in the right places all the way down. And the histone groups find those places by, you know, this frenzy of the water molecules and the other molecules heat, thermal energy. Another just comment on that. You know, humans, we live at some 37 and a bit degrees Celsius. And if we go a little hotter than that, like even 40, 42, something like that, we're roasting. We basically the oscillations get so strong. Maybe they're at 1600 or 1800 miles per hour, and they start causing tremendous damage, and we die. And yet, if we go a little slower, let's say 36 degrees, 35 degrees, the enzymes can't function rapidly enough, and some maybe don't function at all, and we start to slow down, and we get lethargic, and our brain doesn't work, and we go into kind of like a stupor. I forget what they call that, but what are they? It's some kind of exposure. Anyway, it'll come to me. But we go into a coma and then die with just a small lowering. So the thermal activity is actually very germane and very critical to our metabolism, our ability to function. So let's go on here. So here we show the let's read the title. Open access article, Molecular Evolution of the 10-11 Translocation. So that's that Tet word. I think I call it transfer, but it's actually translocation, which is a very similar idea. Gene in mammals. Tet1 and Tet2 are enzymes for the corresponding Tet1 and Tet2 genes. The genes, Tet1 makes the Tet1 enzyme, and the gene Tet2 makes the Tet2 enzyme. And okay, so we're looking at the we're looking at cartoon diagrams, but good ones. Cartoon diagrams for the Tet2 enzyme in this particular case. And we see here this one with all the green spirals. This is showing like a skeleton diagram. It's like looking at a human skeleton. But it's not showing the flesh, the surface. And in these molecular sense, the surface is the electron contour, positive and negative charges. And the contour is the amplitude, whether it's strong charge or a weak charge, creates the hills and valleys. And there also is a spatial location close to the actual atoms themselves. So it's like putting this flesh on the skeleton here in this illustration B. Now I want you to just look at that shape. It's quite rememberable, and you'll see it in the movies. People tend to use this image quite a bit. So just remember that in particular is the Tet2 gene. Oh, this is the Tet2 enzyme. And the product of the Tet2 gene. And the other ones, the other Tets may look somewhat similar, or at least reminiscent to this one. And now it's time to show you a movie. Let's have some fun. Now I want you to remember, just before we go into this, remember Oct4, that octopus you fought. Now remember the 2 socks you put on you felt remorse, and then remember the remorse made your heart feel like it was tearing apart with a jagged cliff between it. Cliff with a K, so it's cliff, and it had 4 like the saw, because it was from the Oct4 tragedy where you had to chop their legs off to protect yourself. And so remember that's the Oct4 SOX 2 and the KL Cliff F4, Cliff 4. Let's just remember those as we go into those crazy images, and watch this absolutely astonishing video.
In order to create induced pluripotent stem cells, a large population of reprogrammable somatic cells is required. Many somatic cells have been successfully reprogrammed. However, fibroblast cells are commonly utilized because they can be easily extracted from a patient using a safe and non-invasive skin biopsy and are easy to culture in a lab. Fibroblast cells are somatic cells that produce connective tissue. Morphologically, the cells appear flat, and they secrete collagen and elastic fibers that make up the extracellular matrix.
Protocol.
Every cell type in an organism, apart from gametocytes and undifferentiated stem cells, are somatic cells. Each somatic cell has a unique structure and function due to a distinct combination of genes that the cell actively transcribes and translates into functional proteins. The difference in gene expression between different somatic cells is largely due to the structural organization of the chromosomes in each cell. Chromosomes can exist in 2 forms. The DNA can either be tightly wound around histone proteins to yield heterochromatin, or the DNA can be loosely unwound to form euchromatin. This loosely unwound DNA permits the binding of transcription factors and other transcriptional machinery, such as RNA polymerase, to the gene's promoter region. Once the transcriptional machinery has bound, gene transcription begins, and a molecule known as messenger RNA is made. When the DNA exists in a tightly wound configuration, the DNA is not exposed to the transcription machinery, and transcription does not occur.
It's unwinding.
In this manner, the cell can regulate the genes it does and does not express. In fact, out of the 20 to 25,000 genes of the human genome, a somatic cell will only express a small fraction of the genes at any given time, and the rest of the genes are considered transcriptionally silent. This is the case for the reprogramming factors OCT4, SOX2, KLF4, and c-Myc in the fibroblast. Because these genes are not actively expressed, scientists must induce the expression of these genes artificially using transgenic strategies. There are a number of methods available for artificially overexpressing genes in a cell. The Yamanaka group utilized retroviral vectors as the method to deliver the transgenes encoded for the 4 reprogramming factors OCT4, SOX2,
There we have it.
KLF4, and c-Myc.
Last one we don't use.
The retroviral vectors deliver the 4 transgenes into the cells. The transgenes are then integrated into the host's genome, thereby permitting its long-term gene expression. Amazingly, when the 4 transgenes are co-expressed in the same somatic cell, the differentiated cell is reprogrammed into a pluripotent-like state.
We don't want that pluripotent state. And we used the 3, OCT4, SOX2, and KLF4, but not the CY genes. We used just the first 3 in the OSK protocol. So there, that wasn't so bad. Very good to see it in action. So now I'm into the type portion of the Dr. Sinclair's paper that I've paraphrased, and it's, like I said, like code, so I have to read it carefully in order to get it right. So here we go. Information from what might soon become the paramount discovery in the history of science as recorded in the paper. And I'll repeat the paper again. Reversal of aging and injury-induced vision loss by the Tet-dependent epigenetic reprogramming. So again, my purpose here is to shed some light on the paper and to expound on the immense implications of Dr. Sinclair and his team's work. As a first test of the hypothesis that mammalian cells might retain a faithful copy of the epigenetic information from earlier life, the Harvard team introduced the expression of the 3-gene, that's OSK, we talked about that, combination in fibroblasts from old mice, and measured its effect on the RNA levels of genes known to be altered with age, including H2A, H2B, lamin B1, and CHAF1B. Now, we don't have to remember that, but there's 4 genes that are known to be altered with age, and they're going to check that. A fibroblast is a type of biological cell that synthesizes the extracellular matrix and collagen and produces the structural framework for animal tissues. In my case, that produces the crow's feet and a little bit of crumpling and all that we get. And for structural framework, collagen and in animal tissues, and plays a critical role in wound healing, where the collagen has to form the fibers again in the tissue, and it plays they excluded the c-Myc gene from these experiments since it is an oncogene, which means it causes cancer. Remember I said uncontrolled cell growth, hair, and teeth. And that reduces lifespan. So we don't want the c-Myc basically for 2 reasons. One is we don't want that uncontrolled cell growth, and we don't want also to take the animal right back to the embryonic stage. You want to just go to youthful age, not right back into the womb. So they found that the OSK treated old fibroblasts, like you saw in the movie, promoted youthful gene expression patterns with no apparent loss of cellular identity. So they didn't, with the 3-stage process, not 4, 3. With a 3-stage process, the OSK-treated fibroblasts promoted youthful gene expression patterns with no apparent loss of cellular identity and without the induction of NANOG, an early embryonic transcription factor that can induce teratomas, and these are strange proliferations of tissue, often including hair and teeth, when present. They thus succeeded in demonstrating that the OSK protocol successfully produced more youthful gene expression in mouse fibroblasts in vitro, that is in the test tube type environment, not in the living animal yet. Notice here I put this heavy arrow to the, well, look, to the right. Every time there's a conclusion, so you wake up there and you can, if you do a screensaver or something, you can read quickly what the conclusions are, what the actual, the discoveries through this paper, and there are so many discoveries. Okay, next. The next step was to test the OSK protocol, and you should know what that is by now, in a mouse tissues in vivo, that is in living mice. The team deployed adenovirus-associated viral vectors, that's AAV, I also sometimes say attenuated as well, because they are attenuated adenovirus vectors, including tetracycline response elements, TRE, to provide tight control of the gene, specifically the ability to turn the genes downstream of the inserts. So they're putting in these vectors, and inside that they have on and off switches, molecular ones, that they also insert, so that they can turn the genes downstream of the inserts either on or off. So they put a switch in front of these genes, the Tet1, Tet2, so that they can turn them on and off, and they can basically make the animal or the cells in vitro and or in vivo, make them go younger, and then they can age, and then they can put in the tetracycline again, which turns the switch on again, and then as long as they keep giving them the tetracycline, I think it's oxycycline, the actual name of it, brand name, keep giving that to the animal and keep it on as long as they like. And then when they stop giving it, the switch goes back off, and those gene inserts do not produce the Tet1 or 2 enzymes anymore. So it goes back to the normal kind of without any intervention metabolism of the cell or the animal. Okay, so let's go to this next one. Now again, I'm going through the paper, paraphrasing it very, very carefully. The control sequence with minimal promoter is called tetracycline response element, TRE, because it responds to the binding of the tetracycline transactivator protein, TTA, by increased expression of the genes or genes downstream of its promoter. In other words, it's like a light switch, on, off. When you give it the tetracycline, it goes on. When you take the tetracycline away, like don't put it in the water anymore, the switch goes off. The control sequences with a minimal promoter is called a tetracycline response element. Okay, we got that. The TRA promoter can be activated with either by the RTA in the presence of tetracycline, that's the Tet-on system, or by the TTA in the absence of tetracycline, the Tet-off. So the Tet-off is like the default state of that switch. So by leaving it without the tetracycline, it's off. Put the tetracycline in, it's on. So you have control now. Within the animal, without doing anything but adding tetracycline in the water whenever you want these genes to be activated. Very cool. Extraneous AAV sequences, remember the attenuated adenoviruses, were removed so the vector could accommodate the OSK as a polycystron. Some people love big words. I don't like it so much. It's like polycystron just means multiple genes, and in this case, as you will talk later about, you put them in series, like three, those trucks where you put the what do you call them? Let's say the cargo portions. You put them in series. They look like a train when you're done. In tandem. Okay, now here comes the exciting thing here, beginning of the excitement. to test if the ectopic OSK, ectopic meaning the outside control of the cells or the animal, was toxic in vivo, that's in the animal. They infected 5-month-old, some kind of mouse, I don't care, with the adenovirus 9 RTA. So they're telling you exactly how they did it, what they exactly used. And the adenovirus with that TRE, the on switch, OSK, and you know what that means but now. That's the OCT4, SOX2, and the KLF4 OSK. So they're giving the adenovirus without the inserts, and they're a control. But you have to give them something in the control as well to make it sort of a perfect match to show that it's the OSK that's doing it, not the attenuated virus. So they gave the virus in its native form. And then they gave the virus, the adenovirus, with the TRE-OSK into the second group of mice, and that's the experimental group. So they delivered it intravenously and then induced the OSK expression in one group by providing doxycycline, the turn-on switch, in the drinking water. Now, surprisingly, continuous induction of OSK, they did it for a whole year with the doxycycline in one of the groups. They gave it for, well, actually, I think they gave it to both groups because it didn't matter in one group. So they have to keep everything in, what's the word, balanced. So that it'll have a test significance. So they have to give it to both groups. So surprisingly, continuous induction of OSK for over a year had no discernible negative effect on the mice. And they say ostensibly because of avoidance of high-level expression in the intestine, thus avoiding the dysplasia and the weight loss seen in other studies. So with that particular protocol they used, with the 3, not 4, genes in tandem, they were able to have a high level of expression and no negative effect. That makes this look very promising for humans, because if we're like the mice, we could take this for a whole year and not have any negative consequences. It's amazing. So the optic nerve is where the retinal ganglion cells, which they're abbreviating RGCs, retinal, the back of the eye, ganglion cells on the retina, bundle together and project towards the brain, where they contact and make connections with the brain. So you get vision, the mice can see. Early during embryogenesis, that's when in the development of the mammal in this case, in the embryo, and also for a while as neonates, so the young baby mice, and even humans have some of this. The RGCs, retinal ganglion cells, can regenerate if damaged, but this capacity is lost within days after birth. Throughout adulthood, the functioning of the RGCs continues to decline. And up until the time of these experiment attempts to reverse the acute or chronic damage to the CNS, central nervous system, had been moderately successful. So up until the time of these experiments, so that's until now, the success they had was when you were looking at the reversing acute or chronic damage in the central nervous system. They were moderately successful in like medicine, doctors, in reversing a lot of this, because the brain has the ability to rewire. Yet, this is the important part, yet no treatments had been, up till today, been able to restore eyesight once the optic nerve connection from the eye to the brain had been so damaged as to produce blindness. Now, this occurs in, for example, glaucoma in humans. So apparently well, we know that until Dr. Sinclair's experiments, there had been no protocol that could heal the optic nerve and reverse blindness from optic nerve damage in the axons. But this protocol could do it. So let's talk about that more. To test whether or not it's possible to restore regenerative capacity in the RGCs, the retinal ganglion cells, Dr. Sinclair's team enacted the OSK protocol. We know what that is. Remember OCT4 and octopus with 4 legs. This SOX2 and the KLF4 from the, yeah, you remember the picture. Protocol mice whose nerves had been crushed earlier. So they crushed the nerves and then later gave the mice, the optic nerves they crushed, gave the mice the OSK protocol. The Tet off demethylation turn off system carrying OSK either in separate AAV vectors or in the same AAV vectors was injected into the vitreous body liquid interior of the eye, resulting in efficient, selective, and tetracycline responsive gene expression in the RGS. It says figure, I'm going to take that out. I don't need that here. I'm not doing Dr. Sinclair's figures because I'm using other figures. So again, I do recommend you read this marvelous paper by Dr. Sinclair. And preferably for me, after you watch this, so it's a lot of the things that are not explained in the Dr. Sinclair's wonderful paper are explained in this, and there different things are explained in Dr. Sinclair's paper. More detail into the protocols and more images in the figures, that sort of thing. So again, I'm coming back to what I just said about the protocol. They injected the same AAV vectors in the 2 mice groups, but in one of the mice group they got the OSK, and the other mice group they did not get the OSK. Okay, as a negative control, a second group of mice were provided the same Tet off protocol, and in addition were continuously treated with doxycycline to replace OSK expression. So they gave the same Tet off protocol, but not the SOX, the OSK protocol. Remember the OCT4, the SOX2, and the KLF4. They didn't give them that portion, but they gave them the TRE sequence, the on and off switch portion. So they're keeping everything as balanced as possible, but so that they can just test the OSK protocol, and everything else is balanced between the 2 mice groups. So the team found that the greatest extent of axon regeneration and retinal ganglion cells survival, RGC survival, when all 3 OSK genes were delivered concurrently in tandem via the same attenuated adenovirus vector, IG as a polycystron, 3 genes in series. So they found that that's what produced the greatest extent of axon regeneration. So that's very promising. This regeneration was for pre-existing RGCs, not to be confused with new RGC proliferation. So what it did, the OSK, was greatly help to stop the RGCs from dying, the retinal ganglion cells in the back of the eye, from dying from having their roots crushed. When you gave the OSK protocol, not very many of them died. But what we're saying here is that none that they could tell grew new ones either. It wasn't a reproducing and proliferation of the RGCs. It was that they healed themselves. They were able to regrow their axon, in the optic nerve, the bundle of axons. When the genes were co-delivered by separate AAVs, so you put, rather than putting them in tandem, 1 to 3, in the same attenuated adenovirus, if you split them up and gave one of those, the Tet1, let's say the what is it, SOX2, well, OCT4 first. You put the OCT4 in one adenovirus bunch, and separately you put the SOX2 in another bunch of adenovirus, and the KLF4, and then yet another group of viruses, and then give all the 3 viruses with the 3 separate ones. They were able to increase the RGC survival slightly, but none of the single factors alone had any effect on regenerative capacity. So what this says is you need to put the 3 factors in the OSK protocol, the 3 genes, in tandem within the same attenuated adenovirus, okay, and give them a bunch of those viruses. Now the reason is what that means is they need those 3 genes need to work together, and to work together they need to be spatially congruent, in other words, together, and temporally together. So you have to putting them in tandem on one strand is a very important distinction. You just can't put them on separate ones and put them all in there and expect the thing to work. They need to work together in space and time. Okay. Said that. When delivered singly, the OCT4, the SOX2 alone increased RGC slightly, but none of the single factors alone had any effect on regenerative capacity. Because KLF4 had been reported to repress rather than promote axonal growth, they also tested a dual system tandem of just OCT4 and SOX2, but observed no regenerative effect, even in the absence of KLF4. Okay. And here I make the conclusion. Thus, with that right-facing arrow that I talked about in bold, thus, in a brilliant piece of deductive scientific work, they were able to determine that all 3 genes, OCT4, SOX2, and KLF4, are required to be introduced spatially and temporally together in order to function so as to restore regenerative capacity in adult RGCs. So here they're telling us that they have a very promising protocol that works in mice and might work in humans to repair the damage induced by damage in the optic nerve, such as in glaucoma. So what this promises, I haven't delivered it yet, but it's promising, is a cure. Maybe not 100%, but you'll see a significant curing of glaucoma. So this is astonishing. Okay, so let's continue on. Utilizing the Tet1 adenovirus system for its rapid on rate, they tested the effect of inducing OSK expression before or after optic nerve crush damage. So they want to see if they give the OSK before and then crush the nerve, the neurons. What does that help it? Or what if we crush the nerve, optic nerve, and then after give the OSK protocol? Does that work? How do those two protocols, experimental protocols, differ? So what they found was significant improvement in axon regeneration was observed only when OSK expression was induced after the injury. Furthermore, the longer the duration of OSK induction post-injury, remember, you can just keep giving them the doxycycline in the water, and those genes will stay turned on doing the OSK expression, right, for as long as you like. Furthermore, the longer the duration of OSK induction post-injury, that's what that means, keep it turned on, the greater the distance of the axons extended. Holy cow, the longer you give the OSK, the better the results. Again, they found no increase in the total number of RGCs. So the retinal ganglion cells are not undergoing meiosis and producing more ganglions. No, they're just healing the ones that were there at the beginning. A second discovery of many. I'm not going to put numbers on them. Now let's just say next, because there's so many. By comparing infected RGCs numbers pre and post injury, they determined that the survival rate of the OSK infected RGC was 2.5 to 3 times greater than that of the uninfected or GFP infected. That's the one without the OSK protocol in it, but just having the on switch in it. Infected RGCs. So it was 52% versus 17% to 20%. So in other words, the retinal, when you crush the optic nerve and you didn't give the OSK protocol, 52% of the 17 to 20% survived, and when you gave the OSK protocol, 52% survived. So you get maybe half your vision back. Or with the eye, there might be enough redundancy that you get most of your vision back, just to fine details. I can't determine that at this point, but that's what I would expect. So the next conclusion was that the P10 mTOR S6K pathway, previously shown to improve RGC survival, so this is a historical event, and axon regeneration vivo, was not activated in the OSK infected cells post-injury, indicating that a new pathway might be involved. So they're saying not only have they discovered something that does this, RGC regrowth, healing and regrowth, not growing more of them. Not only did they discover that, but it's a new pathway, so which makes it extremely, extremely significant. And you don't have to remember that other pathway. We're not talking about the mTOR pathway. Okay, so now they pondered whether or not neuronal injury advanced epigenomic age. In other words, you crushed the optic nerve. Did that make the cells in that area, the area or the structures have an older epigenome, an older epigenome, more the methylation messed up like in an older cell? They wondered whether the neuronal injury advanced the epigenomic age, and whether the OSK's benefits were due to a preservation of a younger epigenome, given the necessity that the OSK be delivered post-injury, and the known role of the Yamanaka factors, those 3 of them, to reverse DNA methylation age during partially or fully reprogramming in vitro. So they're saying, we think it, based on what we've done in vitro and in vivo, that we expect that it should be making it, it should be reversing the age of the RGCs, the retinal ganglion cells, okay? So to test whether or not the neuronal injury advanced the epigenomic age, the genomic DNA from the FACS isolated RGCs was obtained from the retinas that were intact and also from those 4 days after the axonal injury in the presence or absence of OSK induction, and subjected to reduced representation bisulfate sequencing, RRBS. Now, we don't need to know about that, but if you were in a lab and you wanted to reproduce that, you'd look up what RRBS is and try that out as a test if you wanted to match what, reproduce Dr. Sinclair and their team's results. So that's why that's there. A newly published rDNA clock, which I didn't go into, provided the best coverage of over 72 of the CPG sites relative to other available mouse clocks. So they're going to use that. And here's what the conclusion was when they tested it. They found that its age estimate remained highly correlated with the chronological age of the RGCs. So the test was actually able to tell or confirm what the test researchers already knew, what the age of those RGCs were, the retinal ganglion cells. It's able to confirm. So it's a good test. Consistent with this hypothesis, in the absence of global methylation changes, injured RGCs experienced an acceleration of the epigenetic clock, and the OSK expression using this test, they knew that it proved that it counteracted this effect. So what they've proved here is that the OSK protocol was able to take the RGCs or whatever they were, at whatever age they were, and the damage accelerated that age, and they were able to wind them back in age, do what I like to call a factory reset of sorts. Okay, I want to show here the methyl groups as white spheres bound to the cytosine molecules. So double strand and the methyl groups shown as white spheres, just as a reminder. Okay, so when they talk about demethylation, they're taking those off, the methyl groups, and then as part of the protocol, they have those put back on, but in the right place, you know, using the information that's somewhere in the nucleus. We don't actually know where that information is stored, and even the fact that it is stored is very shocking. It's shocking. It's like you have a giant memory database inside the DNA, or something that effectively operates like that. Okay, to explore reprogramming applicability to humans, the team performed axon regeneration assays on differentiated S-H-SY5Y, just a type, that's what that word is, of human neuronal cultures. And they did that with and without the OSK induction. So right there in red, you can see it. This is testing on human neuronal cells in vitro. I know it's in vitro, which means in this test tube, not in the real eyes of the humans yet. But let's see. If this works on human cells, oh my God, oh my God. You know, let's talk about crow's fever a bit. You could take that skin out, and if it like working on and put it in a test tube and make it young again, and then put it back, for example. I mean, just because test tube, don't dismiss it just because it's a test tube test. You know, this is really significant. Anyway, I'm getting very excited, but let's see what they found. So what they did, the black arrows again, show the conclusions. Look at them. Boom, 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 boom. 1, 2, 3, 4, 5, 6. Six conclusions on this page alone. More to come. So they found that the OSK did not induce human neuronal cell proliferation, which could be a problem if it did, as is similar to what they found for the mice RGCs in vivo. But they're testing in the human ones in vitro, but the mice in vivo. Then they induced axon degeneration using a 24-hour treatment with this nasty substance, vincristine, VCS, a chemotherapeutic agent. So they treated the eye as if they had cancer with this horrible stuff. The cells were then allowed to recover for 9 days. And they measured the DNA methylation age of the induced neurons using a clock for in vitro studies. Okay, so that's actually not actually conclusions. I'm taking that arrow off. Okay, save it. Okay, and here's the conclusion. Next, paralleling the findings for retinal ganglion cells, RGCs, the DNA methylation age was significantly increased after VCS damage of the human neurons. So they confirmed that the neurons not only were damaged, but they became older. Then they found that the OSK, next thing, conclusion, the OSK expression prevented this increase in age, and also restored a younger DNA methylation age without a global reduction of DNA methylation. So it appears so far to be working on the human neuronal cells. Okay, now next conclusion. After 9 days elapsed, subsequent to the induced damage, the neurite area was 15-fold greater in the rejuvenated OSK transduced cells than controls, the neurite area. So there's some growth going on, but it's not proliferation, so it's a healing growth. Next point. In contrast, they found that the recovery from damage was completely blocked by validated Tet2 knockdown. So if you turned off the Tet2 gene, then that's what the knockdown is. It was the recovery from damage was in the human cells was completely blocked by validated Tet2 knockdown, even in the presence of high OSK expression. In other words, if you had the OSK expressed and it would otherwise rejuvenate the cells, but you blocked it, so it's expressed and also blocked. But it was not dependent on the TOR at SK6 pathway was also found, as was also found in the mice RGCs. So they found that the OSK needs both OCT4, and it needs the SOX2, both of them. So you can't just block the Tet2 and expect it to have still work. So these are ways they're going in and trying out different things to quickly get reproduction of the results, but with results and conclusions for humans, just the same sort of results they got from the mouse experiments. Final conclusion. Thus, they determined that the ability of the OSK to reprogram neurons and promote axon growth is a conserved DNA demethylation dependent process intrinsic to the cell. So you have to let these OSK demethylize and then remethylize in order to get these benefits. The axon growth in this case. They refer to this process as the recovery of information via epigenetic reprogramming, or reviver for short. I'm sure they were all tremendously surprised when they found that what they named it, the recovery of information via epigenetic reprogramming, just happened to, when concatenated, produce reviver. What an astonishing coincidence. Anyway, let's continue on here. So now let's talk about glaucoma. Glaucoma is a leading cause of age-related blindness worldwide, involving progressive loss of the retinal ganglion cells and their axons. Glaucoma is also often associated with increased intraocular pressure, the fluid in the eye, pressure increase, but it may occur sometimes even in the absence of high intraocular pressure. Present treatments can often slow the progression of vision loss, but it is not possible to restore vision once it has been lost up to now. Now, I need to put that up to now in there as well and emphasize that. Up to now, but we're very, very excited about being able to change that soon. Okay. They tested whether the recovery of information via epigenetic reprogramming, reviver, they're calling it now, could restore the function of RGCs for a chronic disease like glaucoma. So they're not ready to test it in humans yet, because that wouldn't be ethical at this point, or legal probably. So what are they going to do with mice in order to test this? Let's find out. They introduced elevated intraocular pressure, we're calling IOP, in one eye for 4 to 21 days by injecting microbeads into the back of the eye. So if you have a confined cavity in the eye and you put things into it like rocks, little rocks, it causes increases the volume inside the eye of material, and it increases the pressure. So as the volume of the material goes up, the pressure goes up. The volume of the material, not the volume of the eyeball. Okay. The AVVs and or PBS were then injected intravitreally, literally injected into the interior of the eye. They removed the microbeads at a time when glaucomatous damage was established. A lot of these words I've never had to say them, so even though I read them, it's kind of an interesting experience. Glaucomatous damage was established with a significant decrease in RGCs and axonal density. So that they removed the beads once they had established a significant decrease in the RGCs through maybe death, partial death, and axonal density. So the axonal density, some of the axons are dying off, and the density is going down. These are sick RGCs. 4 weeks after adenovirus-associated injection, the OSK-treated mice demonstrated a significant increase in axon density compared to mice that received either PBS or AAVs with no OSK induction. So the OSK-treated mice got a significant increase in axon density. In other words, the existing RGCs, retinal ganglion cells, or ganglion cells, retinal ganglion cells, they grew longer axons only when they got they increased the axon density. The increased axon density was equivalent to the axon density in the saline only non-glaucomatous mice, not the mice without having the glaucoma, and was not associated with the proliferation of RGCs. So you didn't get uncontrolled cell growth. You got, may I say, controlled cell repair, and then damaged cells, damaged human cells. It's like, oh my heavens. This would indicate that the existing RGCs regrew their axons, the ones that were damaged. Oh, that's actually my paraphrasing, I think. I like that much better. It's so much easier to say, and I'm sure it's easier to understand. So, to cure glaucoma, it is not only necessary to regenerate the axons as they had achieved, but it is also necessary to see if the axons regenerated were able to connect with the brain in such a manner as to re-establish functioning vision. So the real test is, can the mice or humans, we can't do the full test in humans yet, but do the OSK mice, actually see again? So the team demonstrated that, this is the result here, that the increased axon density in OSK-treated mice actually coincided with increased vision. They tracked the visual acuity of each mouse by measuring their optomotor response to visual stimuli. Contrary to mice that received either the PSK or the negative OSK AAV, in other words, where they didn't give them the upregulation, in other words, left the switch in the off position, those that received OSK induction with the switch turned on experienced a significant increase in visual acuity relative to the pre-treatment baseline measurement. So they got significantly better, and they saw by testing their sight, they actually saw significantly better. In the OSK-treated mice, about half of the vision was restored. Half of the vision restored in the OSK-treated mice. They recorded the electrical wave generated by the RGCs in response to a reversing contrast checkerboard pattern, and demonstrated that the OSK treatment significantly improved RGC function relative to the pre-treatment baseline measurements. And the PBS or the OSK AAV, in other words, the OSK treatment, but with the switch left off. As far as the team could ascertain, their reviver process is the first treatment to reverse vision loss in a glaucoma model. Wow. The first time they've reversed vision loss in mammals. The team wondered if reviver might be, that's the OSK protocol, might be effective in not only in young mice, as they had achieved, but also in aged mice as well. To test this, the same nerve crush injury and procedure was induced on 12-month-old mice. So this, we're talking, if this transfers to humans, we could apply it to elder individuals who have a much larger likelihood of having and suffering from the effects of glaucoma. So this is a key measurement here. The team ascertained that in aged mice, the OSK with the AAV treatment for 2 weeks after the injury doubled the RGC survival from the nerve crush damage, similar to what they found for 1 and 3-month-old mice, with the exception that the axon regeneration was only slightly less than young mice 2 weeks after the injury. So it wasn't quite as much. But in a follow-up, the OSK AAV treated mice after 5 weeks produced similar axon regeneration to that observed in the young mice. So just leave the on switch turned on for 5 weeks rather than 2 weeks on the elderly mice. It's no big deal. And you catch up. You produce the same visual enhancement that you get in the young mice. Wow. They thus showed with the data that the OSK AAV treatment was almost as successful for producing axon regeneration in aged mice as in young mice following the optic nerve crush procedure. So you can apply it to young and old. Wow. To cure glaucoma, it's not only necessary to regenerate axons as it had achieved, but it's also necessary to see if the, okay, so I think this, yeah, this is a repeat. Great, I can get rid of it. So, okay, so this is the next one here. Having proven, I wonder if that was a repeater I just got. Yeah. Perfect. The team, okay, having proven the ability to recover from crushing injury, next the team tested whether the mice using Reviver were also able to recover from damage to vision due to natural aging alone. So humans don't have to, and mice, don't have to have actual glaucoma to have the eyes get weaker. What is it? Macular degeneration, all sorts of other things. The neurons can just sort of degenerate on their own through aging alone. The team had already determined that there was a significant reduction in visual acuity and RGC function at 1 year in each of these mice as compared with 4-month-old mice, as measured by OMR and PRG. Now, you could look that up. I don't even know what it is. It's not relevant to what we're doing here today. The team gave 4 and 12-month-old mice in intravitreal, like in the eye, injections of negative OSK or plus OSK AA adenovirus treatment. So, you know, in one, they balanced it. They got the same treatment, but in one group of mice, they didn't turn it on by giving them doxycycline, and the other group, they did turn it on with doxycycline. The team found it striking that the age-induced vision loss was completely restored by 4 weeks of OSK expression in both the 4-month and the 12-month-old mice. In other words, oh my God, you know, the expectation, and that is not, I mean, I'm happy about mice being able to see again, but what really makes me excited is this promises if the same results apply to humans, you can give it to young people and older people who've had vision damage through normal, means, and their vision could be brought back to the vision of a youthful person, completely restored. That's what that means, completely restored. Wow. In contrast, for yet older 18-month-old mice, remember we did 4-month and 12-month, 18-year-old mice did not find a commensurate increase in vision using the same procedure. But this is likely because the 18-month-old mice had cataracts, and these cataracts did not clear using the OSK protocols. So, phew. Yeah, if the cataracts cleared, they could see, but they didn't clear. So that's probably what that was. In these experiments, the degeneration of vision was not associated with much axonal loss. The team hypothesized that the loss must be due to loss of function rather than loss of axons in the RGCs. Comparing the RGCs from 5-month-old young mice, the team identified 464 genes that were differentially expressed during aging and also not affected by the empty AVV. In other words, not turning it on. So they give them the protocol, and if they don't turn on the OSK genes with the doxycycline, these were not affected. Okay, great. Remarkably, though, the vast majority, 90% of that's 418 of the 464 age-deregulated genes, were restored to youthful levels after 4 weeks of OSK expression. So they're demonstrating here that in the mice, 90% of those the signs of aging were reduced, reduced to being at the youthful levels, restored to youthful levels after 4 weeks of OSK expression. Okay, so of the 268 age downregulated genes, 44 were genes involved in the sensory perception, suggesting a decline in signaling receptors or sensory function during aging. One that can be restored by reviver. Wow. Interestingly, 116 of these genes are yet to be characterized. Another 196 of the genes that were upregulated during aging are known or predicted to be involved in ion transport. So these are right into the technical minutiae, but it looks very, very promising, restoring age damaged RGCs to youthful levels. Now, maybe not 100%, but close. Okay, so remember that the Tet1 and Tet2 upregulate the necessary demethylation operation necessary for reprogramming. So in order to fix the problem where these methyl groups get out of place, you first remove the methyl groups, and then you do the factory reset operation with the other gene enzymes and put the methyl groups back through some as yet mysterious process, like a factory reset using memory somewhere in the DNA. So consistent with this fact, the axon regeneration data showed that the knockdown of Tet1 or Tet2 completely blocked the ability of reviver to restore vision in 12-month-old mice. Now, I'm going through here the conclusions. So that's conclusion 1. To determine if the DNA methylation clock was affected, Dr. Sinclair's team measured the rDNA methylation age of RGCs from 12-year-old mice. Okay. They found that 4 weeks of having the OSK AAV with the expression turned on significantly decreased DNA methylation age. And this was, they also found, proved, Tet1 and Tet2 dependent, so the two have to work together. Together, these experiments demonstrated Tet-dependent in vivo reprogramming that restored youthful gene expression patterns comma, reversed DNA methylation clock, and restored the function and regenerative capacity of a tissue as complex as the retina. Now, the retina is not like a skin cell or a toenail cell. It's a retinal cell is one of the most complicated and differentiated cells in the human body, if not the most complicated. So to be able to achieve this regeneration for this particular type of cell is extraordinarily significant and extraordinarily promising. This regeneration was achieved and hence proven to be achievable even in the post-mitotic neurons in the central nervous system that are some of the first cells in the body to lose their ability to regenerate, and hence are some of the cells that are more difficult in which to achieve such regeneration. So Dr. Sinclairs and his team at Harvard didn't take on the easiest problem first. They took on one of the hardest problems to really prove their case, which they did. Oh my God. In this next one, in this study, they showed that in vivo reprogramming of aged neurons did and hence can reduce DNA methylation age and allow the aged neurons to regenerate and function as though they were young again. Wow. Next one. The requirement that the DNA demethylases, yeah, Tet1 and Tet2 be active for this process indicates that altered DNA methylation patterns may not be just a measure of age, but that the DNA methylation patterns themselves are likely to be active participants, in other words, causation agents in the production of aging. So what they're saying here is we've demonstrated that these methylation patterns are causal. They participate in the aging process itself. So they're getting at some of the root causes and reversing them. This is fountain of youth type talk, and they've demonstrated it scientifically. So this is an amazing, amazing results. And the last two conclusions. These data led the team to conclude that mammalian cells retain a set of original epigenetic information. The information is there somewhere. We don't know exactly where or how yet, but we've proven it has to be there. We know it has to be there, and that it's accessible and that it works. You're able to do a factory reset. And then he's speculating in the same way that Shannon's observer stores information to ensure the recovery of lost information. Now, I happen to have a master's in electrical engineering, and I studied communication theory in great depth. So I understand Shannon's, what the Shannon's approach was. So using Shannon's, I'll give you an example. Using Shannon's approach, redundancy can be built into the information according to pre-specified rules known to the data reader, okay? Each redundant sequence could have a checking system, such as checking the sum, the digital sum, 1 and 0, 1, 0, add them up. And there's certain rules that you can check to make sure the sum is correct for the size of the message. This could allow the receiver to detect an error and to perhaps fix it. And if the error is not fixable by a sum check, then the sequence could be restored by copying the sequence from a redundant portion. Remember I said put redundancy in there, of the sequence that did pass the parity check. So this is just an example of the sort of things that are done in communication theory, using communication theory to allow us to send messages and repair and detect and repair bit changes. Such as when you watch me right now on this video, and one of the things that allows you to see it without the glitches and the tilization is they do error correction code. So what he's saying is there has to be some kind of error correction code. Not that it's using the Shannon's approach, but it just has to be similar in some respect. Having redundant information, having a way of coding it in advance so they can detect when it's not correct and it needs to be fixed or put back or some such thing. So exactly how this sort of redundancy coding into the methylation is used to retain youthful DNA methylation patterns, and then in adulthood using OSK to instruct the successful removal of deleterious marks is unknown. So I know it's a mouthful, but they're just saying that exactly how this information is accessed and deployed in the OSK protocol is not understood yet. We just know how to make it work. It's experimental and we know how to use it, but we don't fully understand it yet. So how that works remains a subject for further inquiry. And this is a major deal. This is so exciting to look into, as well. Okay, here's the last point. I'm going to, get rid of this by doing ignore. There we go. Let's being stubborn. We're going to stubborn. There, we did it. Okay, even in the absence of this knowledge, these data indicate that the reversal of DNA methylation, age, and the restoration of a youthful epigenome could be an effective strategy, not just to restore vision, but to give complex tissues the ability to recover from injury and to resist age-related decline. And this applies in mice, and it's very promising that it may also apply in more advanced mammals such as humans. Now let's look at this paper is so amazing, so apocryphal. It's just immensely important. And so I want to at least talk about some of the future implications and questions. So this is, I'm not, it's a word, I'm not praising Dr. Sinclair's work, but at this point, this is some of what I've been thinking or what comes to me. Possible future developments and questions. 1. Tests for the efficacy of epigenetic reprogramming to treat human glaucoma. Okay, Dr. Sinclair is planning that in the next 2 years or so. Incredibly exciting. So this doesn't come from me. To better as a better alternative to blindness. I'm saying because with human glaucoma, you can inject it in the eye. It doesn't affect the rest of the body, perhaps at all, and you're basically the options that the person with the glaucoma who would participate in these trials, would likely be advanced in their choices to go blind or to try this. So it's a very ethical group to test this in, and a very appropriate disease for which to test this. That's point number 1. Okay, next, primate studies to determine if select primate species close to humans may be age reversed using OSK1 and or a multiple of times. That would be, I think to take, let's say, I don't want to pick a particular species, but let's say you have a very happy but elderly chimpanzee that's otherwise going to die, or maybe has some even diseases or something that would otherwise predispose him early to death. Perhaps it's ethical in those cases to try this out on some of those, reverse some existing. You don't have to wait for them to age. You could try it out on ones now and do it in an ethical manner. That's just me talking. I don't know. I don't want to get in trouble for saying that. But these are speculations and questions. Third one, maximization of life extension effects by coupling with known factors such as the lengthening of telomeres through the stimulation and production of telomerase, and also interventions such as NMN. This is Dr. Sinclair's group's already using that, no doubt testing it, and niacinamide supplementation and other cycle-supporting nutraceuticals. So they've had a chance to test it, this OSK protocol on its own, and there's a whole future of testing it in conjunction with other effects, such as, I mentioned the telomerase and telomere extension, and I just want to mention astragalus is a well-known herb has one of the strongest effects on generating or inducing the cell to produce telomerase inside. It's a large molecule, so inside where it needs to be to extend the telomeres at the end of the DNA so that they can reproduce longer, or maybe I'd hope indefinitely, so that the cells can support life without basically falling apart in their structure. So I'm saying there's a whole rich area of exploration and how you can use the OSK protocol in conjunction with other therapeutic approaches. Okay, using these methods, can we extend the wild-type genome animals' healthy lifespan to arbitrary lengths? So, there are laboratory or bred for laboratory types for mice and rats that have a reduced lifespan, and they can demonstrating now that they can expand that lifespan. We all want to know can you take the wild type mammals and extend their lifespan, and how long can you extend it? And if they die suddenly after a life extension, let's say, healthy animals just suddenly die. I don't know. I've heard that from Sinclair himself. Can we do things? Why? Why do they die at that point? Is it through telomeres getting too short, or what's the mechanism? And can we fix that? So these are questions. Also, can the epigenetic reprogramming cure cancers by resetting the developmental clock? So if you take cancer cells and do OSK on them, can you, like some of the cancers are caused by methylation changes, if you ran them back in time to a youthful level, does the cancer go away? So there's another vast field of study. And lastly, but not least at all, I'm going to finally say it. Can OSK be safely given to humans to extend their lifespans as a one-time or perhaps a repeated process? Can we extend the healthy lifespan of humans and perhaps indefinitely? Like this world is so incredible, and there's so much to learn. I'm sure I'm not the only kind of scientific mind and researcher that finds that their life is running out, and there's so much more to learn, and you're still mentally and thank God, in my case, physically at this point, still very, very young. but boy, I can't imagine anything more exciting. Well, at this point, to being able to extend the lifespan of not only yourself but your family, your loved ones. This is powerful. And lastly, I want to redirect your attention to something below there. Now, do you perhaps remember OCT4, SOX2, and KLF4? Will you ever be able to forget those images and what they mean? And when you put them together, you get OSK. OSK, that's the name of this protocol, OSK. And with that, I just want to say, this has been one of the most exciting presentations I've ever made. And this work by Dr. Sinclair and his team is of such a high quality. His paper's so well written. And the import of what they've discovered is so immense. I'm serious, fully serious when I say that this is most likely the most important scientific discovery in the history of mankind. And with that, I'd like to say thank you very much. I've enjoyed presenting this to you so much. And I'm signing off right now. See you next presentation.